Hello students, in this video of chapter at a glance, we will revise the chapter Anatomy of Flowering Plants with respect to NEET examination. Anatomy of Flowering Plants. So if you see the chapter history in the previous NEET exams, the average number of questions that appear in this chapter are 2 per year. And you can see the data that is given from 2025 up to 2020. So in every year, you are observing 1 or 2 questions with respect to this chapter. And some of the previous year questions with respect to this chapter, Anatomy of Flowering Plants. So find the statement that is not correct with regard to the structure of monocot stem. So vascular bundles are conjoined and closed. Phloem parenchyma is absent. Hypodermis is parenchymatous. Vascular bundles are scattered. So they are asking which of the following statement is not correct with regard to monocot stem. So if you see here, the hypodermis is parenchymatous is not a correct statement, because if you go to monocot stem, the hypodermis will be sclerenchymatous. Then another question. Bulliform cells are responsible for. So bulliform cells are seen in the upper epidermis of grass leaves. So you can observe the options that are given: inward curling of leaves in monocots, protecting the plant from salt stress, increased photosynthesis in monocots, providing large spaces for storage of sugars. So bulliform cells are responsible for which of the following options is related for the bulliform cells? Then the answer will be inward curling of leaves in monocots with respect to grasses. Then another question which has appeared in 2024 question paper, in the given figure, which component has thin outer walls and highly thickened inner walls? So this is the diagram of a stomatal apparatus, that of a dicot. So you can see the guard cells which are supposed to be bean shaped or kidney shaped. So you can see that the guard cells are labeled here as alphabet C. So the first option, C, those cells, the inner walls are thick and outer walls are thin. Then the next question. Now these are 2 statements with respect to the 24 question paper. Statement 1, parenchyma is living but collenchyma is dead tissue. Gymnosperms lack xylem vessels, but presence of xylem vessels is the characteristic of angiosperms. So in light of the above statements, choose the correct answer from the options given below. So let us read the statements once again. Statement 1 is parenchyma is living but collenchyma is a dead tissue. Now here you have a wrong concept, that is collenchyma is a dead tissue. Actually the correct combination is not collenchyma. If it is sclerenchyma, it is a dead tissue. Both parenchyma and collenchyma are living. So this statement 1 is false. Then gymnosperms lack xylem vessels, but presence of xylem vessels is the characteristic of angiosperm. Yes, it is correct. Xylem vessels are absent in gymnosperms, but their presence is characteristic feature of angiosperms. So now of these 4 options, statement 1 is false and 2 is true. Therefore, the option is 4. That's one. Then the next one is also a statement type of question from 23 question paper. Endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body. Endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body. Statement 2. Exarch condition is the most common feature of the root system. So in light of these 2 statements, we have to choose which is correct, which is false from the given options. So endarch and exarch are the terms often used for describing the position of secondary xylem. No, they are not used for the secondary xylem, they are used for the primary xylem. So statement 1 becomes false. Exarch condition is most common feature in the root system. Yes, that is correct. Where protoxylem is towards periphery, metaxylem is towards center, therefore it is exarch. So here the option is statement 1 is not correct, but 2 is correct. Therefore it goes to the second option. Now you can see another question, that is identify the correct statements. In this, the first statement is lenticels are lens-shaped openings permitting the exchange of gases. That is first statement. Second, bark formed early in the season is called hard bark. The third statement, bark is the technical term that refers to all tissues exterior to vascular cambium. The next is bark refers to periderm and secondary phloem. The last E, phellogen is single layered in thickness. So we have to choose here the correct answer, taking which of the above statements after reading A to E, which statements are correct. Now, lenticels are lens-shaped openings permitting exchange of gases is correct. But bark formed early in the season is called hard bark is wrong. That's not correct. The term hard bark is not correct. Then bark is a technical term. Technical term is wrong. It's non-technical term. Then bark refers to periderm and secondary phloem. This is correct. And phellogen is single layered in thickness. No, it's more than one layer. Therefore, the correct statements are A and D. That is fourth option is correct. After going through the PYQ questions, now let us go through the main points of NCERT textbook with regard to this chapter. Now you find that similarities and variations are seen in plants as well as in animals, both externally and internally. So externally already you have observed the variations that are observed in plants with respect to the earlier chapter, which is called morphology of flowering plants. So in this chapter, we will be reading about the internal variations that are observed in plants. So the study of internal structure of plants is called anatomy. And what is the basic functional unit in a plant is nothing but the cell. Now, a group of cells which altogether perform a common function will be called as a tissue, and tissues, a group of tissues which perform a common function in plants, we have a special level of organization called tissue systems. Tissue systems, after that at a higher level you will have organs which are like root, stem and leaf. So the special unit of organization in plants in between the cell and the higher level organ is nothing but the tissue system. So mainly in this chapter we will concentrate on the internal structures of typically dicots and monocots, which are part and parcel of angiosperms. But you would observe that within angiosperms, monocots and dicots seen to be anatomically different. So internal structures also show internal structures show adaptations to adverse environments. Now, if you consider the concept of tissue systems, now here the two important points are tissues vary depending upon their location in the plant body. All tissues are not alike. They vary depending upon their location in the plant body. At the same time, the structure and function would also be dependent upon location. So, where is the tissue located? Based on the location, the structure and functional aspect of the tissue is just typically considered. On the basis of the structure and location, there are three types of tissue systems. So what is the criteria for classifying the tissue systems is an important one, that is structure and location. The three types of tissue systems are epidermal tissue system, ground or fundamental tissue system, and vascular or conducting tissue system. So briefly, if we see the epidermal tissue system, it is the outermost covering of the primary plant body, and you can observe that it comprises of epidermal cells, stomata, epidermal appendages like trichomes, hairs, even cuticle is also present. Now epidermis is generally the outermost covering of the plant body, and here you will be observing that the epidermal cells are generally parenchymatous, generally single layered, and they are supposed to be typically having small amount of cytoplasm and also a vacuole. So if you go to organs like leaf and stem, towards the outer edge you will have a layer called cuticle, which is just preventing water loss that occurs by transpiration because it is a waxy layer. But such a cuticle is absent in roots. And stomata are structures present in epidermis of leaves. And stomata regulate the process of transpiration and exchange of gases. So we will discuss about the different components of stomatal apparatus actually. So different components of stomatal apparatus could be like this. Stomata is composed of guard cells, subsidiary cells, and also a stomatal aperture. These are the three components of the stomatal apparatus. So if you just observe, the stomata is composed of two guard cells and the shape of the guard cells will be differing. Each stoma is composed of two bean-shaped cells called as guard cells which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped. So this bean-shaped is with respect to the dicots, dumbbell-shaped is with respect to grasses. And the other one is the outer wall of the guard cells is supposed to be away from the stomatal pore guard cell. The wall which is away from the stomatal pore within the guard cell is thin, whereas the inner walls of the guard cells which is towards the stomatal pore is highly thickened. So now the guard cells are surrounded by specialized cells which are known as subsidiary cells. So the stomatal apparatus is composed of three things. One is the guard cells, the other one is the surrounding subsidiary cells, and the stomatal aperture. Now the other important point is with regard to the epidermal heads. Epidermal heads which you observe on stem, they are called trichomes. They are multicellular. But the extensions with regard to root epidermis, epiblema, you will find unicellular extensions called root heads. So the epidermal extensions with regard to root, they are unicellular and they are called root heads and they help in absorption of water and minerals from the soil. The same in the case of stem, they are multicellular and they are supposed to be involved in decreasing water loss that occurs by transpiration. They may be soft or stiff, they may be even secretory also. Now going to the next typical tissue system, it is called ground tissue system. So all the tissues that are not a part of the epidermal tissue system and vascular tissue system, they come under the ground tissue system. So ground tissue system is supposed to be including all three types of simple tissues, parenchyma, collenchyma and sclerenchyma. And the parenchymatous cells are usually present in cortex, pericycle, pith, medullary rays in primary stems and roots. Whereas in leaves, the ground tissue consists of thin-walled chloroplast containing cells called the mesophyll region. So in leaves, the ground tissue is represented by mesophyll, whereas in the case of the stem and the case of root, the ground tissue could be represented by all the tissues that are not a part of the epidermal tissue system and vascular tissue system. Then the next tissue system is nothing but the vascular tissue system. So if you see the vascular tissue system consists of two complex tissues. What are the two complex tissues in the sense? Phloem and xylem. So based on the presence or absence of cambium, you can observe that the vascular bundles can be divided into two types. If the cambium is present between xylem and phloem, then the vascular bundle will be called as the open type of vascular bundle. If the cambium is not there in between xylem and phloem, then it could be called as the closed type of vascular bundle. So, depending upon the presence or absence of cambium, which is providing an ability to produce the secondary vascular tissues, nothing but secondary xylem and phloem, we can divide the vascular bundles into open vascular bundles and closed vascular bundles. Now, when you consider the types of vascular bundles based on the xylem and phloem, how are they arranged in the vascular bundle? The vascular bundles can be mainly divided into the following types. One is called as the radial type of vascular bundles. The second one is called as the conjoint type. So you could see the first diagram here. This is the radial type of vascular bundle. So you will find the radial type of vascular bundle showing xylem on one radius and phloem on another radius, which you can clearly see in the diagram. Xylem is on one radius, phloem is on another radius. So such type of vascular bundles where the xylem and phloem are arranged in an alternate manner along different radii are present in roots. Whether it is a dicot root or a monocot root, you find the radial type of vascular bundles. In contrast to this, if xylem and phloem are present on the same radius, then it could be called as the conjoint type of vascular bundle, and this conjoint type of vascular bundle could be further divided into two types, namely conjoint closed and conjoint open. The difference between these two is nothing but the cambium. When the cambium is present between xylem and phloem, it could be called as the open type of vascular bundle, or you can also call it as conjoint and open. If cambium is absent, then you have to call it as conjoint and closed. Now here xylem is always present towards the inner side and phloem is present to the outer side in the vascular bundle. So that is one point. Now coming to the transverse sections of the monocots and dicotyledonous plants, we have the explanatory parts for the dicot root followed by the others. Now the first one is dicot root. The outer layer of the dicot root is called epiblema, and it has unicellular extensions called root hairs, which we discussed earlier in the epidermal tissue system, which help in the absorption of water and minerals. And you have several thin-walled parenchymatous cells, which you could clearly observe, and there are a large number of intercellular spaces. And the innermost layer of the cortex is called endodermis. A very important point here that you have to observe is endodermis is single layer. It is made up of barrel-shaped cells, and these endodermal cells on their tangential as well as the radial walls, they have deposition of suberin material, and this is seen in the form of a band which is called Casparian strip. So the radial and tangential walls of the endodermal cells have deposition of water impermeable substance called suberin, which is represented as the Casparian band. Next to the endodermis, you could find the stellar part, which is represented by thick-walled parenchymatous cells, which are represented as the pericycle. So pericycle is a layer from where you will observe the development of the lateral roots. So lateral roots are endogenous in origin because they develop from the pericycle, which is the innermost layer, not like that of the root hairs, which develop from the outermost layer. Now, then during secondary growth also in dicot root, the pericycle cells undergo dedifferentiation and give rise to the cambium, vascular cambium. And the parenchymatous cells which you observe in between the xylem and phloem tissue could be called as the conjunctive tissue. Now coming to the number of xylem strands which you observe here, there are 4 xylem strands, but the range of the xylem strands can be 2 to 4 in the case of dicot root. So therefore it will be diarch to tetrarch, and a cambium is supposed to be developed later during the process of secondary growth due to dedifferentiation from the cells of pericycle as well as the cells of conjunctive tissue with respect to the secondary growth. And now if you just observe the monocotyledonous root, monocot root is more similar to the dicot root. But the prominent difference here is when compared to the dicot root, here you can observe that dicot root has less number of vascular bundles. But whereas here the number of xylem strands are more than 6. So you call it as the polyarch condition. And monocotyledonous root has comparatively well developed pith or medulla when compared to the dicot root. That's the main difference. One is with respect to the number of xylem strands, the other one is with regard to the medullar pith. And in monocots generally you don't find the secondary growth. Now going to the dicot stem, the dicot stem transverse section actually shows the outermost layer is epidermis, which is covered by cuticle. And the cuticle is supposed to be outermost layer, and epidermis has multicellular hairs called trichomes. Sometimes they may be stomata. But below the epidermis, you have the cells arranged in multiple layers. And the first below the epidermis is, the first layer below epidermis is hypodermis. And this hypodermis is made up of collenchymatous cells. So it provides mechanical strength to young stem. And below this you have the general cortex, and which is followed by endodermis. The endodermal cells in the case of the dicot stem are rich in starch grains. So this layer is referred as starch sheath. Now you could see that in the case of dicot stem, you could just observe pericycle is seen as a patch, which is called as the semilunar patch. So this semilunar patch is supposed to be the pericycle. Semilunar patch is the pericycle, which is made up of sclerenchyma. It's not parenchyma or collenchyma, it is sclerenchyma. Now, the radially placed parenchymatous cells, which are seen in between the two vascular bundles, they will constitute the medullary ray. So, what is a medullary ray, if you want to observe, is nothing but this part, this gap in between the two vascular bundles, whatever parenchymatous tissue you see, that is called as the medullary ray, which is the radially elongated parenchymatous cells. Now, in the case of dicot stem, the other important point is you find ring-like arrangement of vascular bundles. And these vascular bundles, which are arranged in ring-like, and their condition is conjoint and open, because cambium is present in between xylem and phloem. So you could find ring-like arrangement of vascular bundles, and the vascular bundles are conjoint and open. That is also a prominent point. And you could also find out that in the case of the xylem condition, it is endark condition. So protoxylem is towards the center, metaxylem is towards the periphery. So which is a centrifugal growth. And we have a large pith or medulla in the case of the dicot stem. The contrast of it is the monocot stem. So in monocot stem, you do not find whatever complete layers are seen in the case of dicot stem. In monocot stem, the monocot stem is clearly represented by mainly epidermis, hypodermis, ground tissue and vascular bundles. Layers such as endodermis, pericycle, even regions like medulla, you don't find in monocot stem. So here the main point is the hypodermis is made up of sclerenchyma, and you observe a large number of vascular bundles in a scattered manner, and each vascular bundle is surrounded by a fibrous sheath, and you have a parenchymatous ground tissue, and smaller vascular bundles you could find are present at the periphery, and the central vascular bundles are somewhat larger. And each vascular bundle has a typically water containing cavity which is called protoxylem lacuna. Now the other part which comes to be the last one is regarding the transverse sections of the leaves. So if you see the dorsiventral leaf, you could observe that leaf being a flat organ will have two surfaces, upper and lower, adaxial and abaxial. So leaf is generally studied under three different regions. One is epidermis, the second one is mesophyll, the third one is the vascular system. So you could now observe that the abaxial epidermis generally bears more number of stomata than adaxial epidermis. So abaxial, which is supposed to be the lower surface, will have comparatively more number of stomata when compared to the upper surface. And the tissue between upper and lower epidermis will be called as the mesophyll. So this mesophyll is well differentiated and it has two types of cells which you could see here. This is called palisade parenchyma and this is called spongy parenchyma. You could see that the cells of palisade parenchyma are supposed to be compactly arranged and they have large number of chloroplasts and their main function is to perform photosynthesis. And this palisade parenchyma is present towards the adaxial surface. Whereas the spongy parenchyma cells are present towards the lower surface and they are somewhat typically having comparatively less number of chloroplasts and there are large number of intercellular spaces between them. So now you could clearly observe a difference in the color with regard to the intensity of the green color, and the dorsal and ventral surfaces of the dicot leaf can be clearly differentiated. So it is called as the dorsiventral leaf. In the case of the monocot leaf, which you actually observe, this monocot leaf, if you just see in monocot leaf, you don't find a clear differentiation of what in the sense mesophyll. So because in monocot leaf, you will be finding either palisade parenchyma or spongy parenchyma. So you cannot differentiate the mesophyll saying palisade or spongy. So mesophyll is well differentiated in the case of dicot leaf, but not in monocot leaf. And similarly, now coming to the vascular bundle, before we go to the monocot leaf, you could see that the vascular bundle, you have phloem towards the lower epidermis, xylem towards the upper epidermis, and each vascular bundle is surrounded by a bundle sheath and there is no cambium. So it is conjoined, closed type of vascular bundle where phloem is present towards the lower epidermis, xylem is present towards the upper epidermis. Because dicot leaves show reticulate venation, the size of the vascular bundles gradually decrease from the base of the midrib to the apex, and even in the lateral veins the same thing is observed. So that is what is supposed to be shown here. You could see the size of the vascular bundles are dependent on the size of the veins and they vary with respect to the thickness of the veins, and each vascular bundle is surrounded by a bundle sheath. Now coming to the point on monocot leaf, as we have discussed, this monocot leaf doesn't have a well developed mesophyll, which is the primary point of difference. It will be either palisade or spongy. And another point of difference when compared to the dicot leaf is the distribution of stomata. Stomata are present on both surfaces of the epidermis. So they are present on both surfaces and there is no irregular distribution. And you could also observe a special point which is not observed in the case of the dicotyledonous leaf. That is, on the adaxial epidermis, you could find along the veins special cells called bulliform cells. There are special cells called bulliform cells. So these bulliform cells, they have absorbed water and are turgid, then the leaf surface is exposed. So when the bulliform cells are full of water, then the leaf lamina will be open. If the bulliform cells are flaccid due to water stress, then they make the leaves to curl inwards, and therefore when the leaf lamina is not exposed to sunlight, the rate of transpiration will go on decreasing. So these bulliform cells, they minimize the water loss, which is supposed to be due to the enrolling of the leaf lamina when the bulliform cells are flaccid. If they are turgid, then the leaf lamina will be open. So these bulliform cells, they are present in the upper epidermis. They can be turgid or flaccid. So with respect to their turgidity and flaccidity, they control the opening and closing of the leaf lamina. and these bulliform cells are seen in the grass leaves towards the upper epidermis. And because the monocot leaves, they show parallel venation, you find that the size of the vascular bundles is almost same, but it doesn't differ like that of the dicot leaves, which shows the reticulate venation. There is a point that could be very important in the case of the summary part, that is, we have three types of meristems which are not generally given in the regular description. One called as the apical meristem, lateral meristem, and intercalary meristem. So these are three types of meristems which are given based on the position: apical, lateral, and intercalary. At the same time, tissues are also divided with respect to their ability to have the power of division in their cells into two types. Meristematic cells having tissues are called meristematic tissues, where the cells retain the power of division, and the cells which have lost the power of division, they become a part of the permanent tissues. So permanent tissues are divided into two types: simple tissues, complex tissues. Simple tissues are made up of one type of cells, which includes parenchyma, collenchyma, and sclerenchyma. Complex tissues are made up of more than one type of cells, which includes xylem and phloem. So what are the functions of tissues? It is given here: assimilation of food and its storage, transportation of water and minerals, and photosynthesis. Mechanical support are the main functions of tissues. And as I told you, on the basis of location of cambium and the xylem and phloem, the vascular bundles could be divided into different types. And you could observe that secondary growth is a process which is seen with respect to most of the dicotyledonous plants in roots and stems, but not in monocots generally. Okay. Structure of stomatal apparatus. Stomatal apparatus is made up of guard cells, subsidiary cells, and stomatal pore. So you could see here guard cells. These are supposed to be, this is the stomatal apparatus diagram with respect to dicots, and this is with respect to monocots. The main difference is with respect to the shape in the guard cells, where here the guard cells in dicots are kidney shaped or bean shaped, whereas here in the case of monocots, they are, especially in grasses, they are dumbbell shaped. And the guard cells are surrounded by the subsidiary cells. These are specialized cells which are seen around guard cells, and each guard cell is supposed to be very peculiar because it is having chloroplast. And the inner walls of the guard cells are thick, whereas the outer walls of the guard cells are thin. When the guard cells are full of water, you could observe that the stomata will be in open condition. If the guard cells are flaccid, they are not having water, then the stomatal pore will be very close. The two walls of the stomatal pore will be closed. Therefore, stoma will be in closed conditions. So, turgidity and flaccidity are the two important points that determine the opening and closing of stomata, which is supposed to be the point that you need to understand. Then the types of vascular bundles. So there are three types of vascular bundles. The first one is the radial type of vascular bundles, where xylem and phloem are present on different radii, seen in dicot root and monocot root. And the second one is supposed to be the conjoint closed type of vascular bundle. This conjoint closed type of vascular bundle, where xylem will be on the inner side and phloem will be on the outer side, and in between xylem and phloem you don't find the cambium strip. Therefore, it is called conjoint and closed. These type of vascular bundles are seen in monocot stem, dicot, and monocot leaf. The third type of vascular bundle is the conjoint open type of vascular bundle. So you could find the difference in these two diagrams very clearly. You have the same position of xylem and phloem, but there is a cambium strip between xylem and phloem, which makes it to be called as the conjoint open type of vascular bundle, which you observe in dicot stem. So these are points regarding the types of vascular bundles. Now, if you see the main differential points between dicot root and monocot root, the difference comes with respect to the number of xylem strands and also the secondary growth points, which are listed with respect to the differences here. You could see in dicot root, the number of xylem strands are 2 to 4, diarch to tetrarch, whereas in monocot it is more than 6, therefore you call it as the polyarch condition. Whereas in the case of dicot root, pith or medulla is absent, whereas in monocot root, pith or medulla is well developed. And you know that secondary growth is present in dicot root, and the cambium develops from the pericycle cells and the cells of conjunctive tissue, both partly are involved in this process, whereas secondary growth is absent in monocot root. So these are the main differences between dicot root and monocot root. Then this is actually the dicot stem, where you can see many points which are very peculiar. One is the epidermis is having multicellular hairs called trichomes, which is followed by hypodermis, which is made up of collenchyma, and you have endodermis followed by what in the sense semilunar patches of sclerenchymatous pericycle, and the vascular bundles are arranged in a ring. Now this is the enlarged view of one particular vascular bundle in a dicot stem. Now if you compare this with the monocot stem, you find here some characters which are totally different from that of the dicot stem. You don't find here the vascular bundles to be arranged in a ring-like manner, but they are scattered. And you are also not finding the multicellular hairs on the epidermis, which are called trichomes. So like this, there are some differences between the dicot stem and monocot stem, which are supposed to be tabulated here. You could see here, epidermis is single layered and epidermal hairs are present. Whereas here epidermis is single layer, but epidermal hairs are absent. So you have hypodermis, which is made up of collenchyma. Here it is made up of sclerenchyma. Vascular bundles are arranged in a ring, whereas here they are scattered. And the vascular bundle size is same. Whereas here larger vascular bundles are present towards the center, peripheral vascular bundles are smaller. And you find that the vascular bundles are conjoined, collateral and open, whereas here they are conjoined, collateral and closed, and each vascular bundle has a sclerenchymatous bundle shape. Another point which is very important here is the vessels are arranged in the shape of Y, and they have a water containing cavity in the vascular bundle. Each vascular bundle has a water containing cavity. So here major space in monocot stem is occupied by ground tissue, which is made up of parenchyma, but here you have a well-defined differentiated layers called as endodermis, pericycle, and even a well-developed region called as pith or medulla. So these are some differences which you could find in between dicot stem and monocot stem. Now in comparison to these two, that is the dicot leaf and monocot leaf, when you identify, you could see the main point here where the difference in mesophyll is clearly seen, which is not observed, and you also have specifically special cells called bulliform cells in monocot leaf, which are absent in the dicot leaf. So those are tabulated here, and you could see more number of stomata on the lower surface, whereas the number of stomata are similar on both surfaces. Mesophyll is well developed here and differentiated into palisade and spongy. It is not well developed, but it is either palisade or spongy. And bulliform cells are absent, but bulliform cells are present here on the upper epidermis. And vascular bundle size is not similar, but here vascular bundle size is similar. That's the point. Now this is the flowchart on the chapter Anatomy of Flowering Plants, which gives you the information regarding different things, that is actually mainly tissues. You could see the tissues are mainly divided into meristems and permanent tissues. Meristems could be primary meristems, secondary meristems based on their origin. Based on their positions, they can be apical, intercalary and lateral. Whereas permanent tissues could be divided into two types, simple tissues and complex tissues. Simple tissues include parenchyma, collenchyma, sclerenchyma, whereas complex tissues include xylem and phloem. Now, if you go to tissue systems, these are epidermal, ground and vascular tissue system, and this is the type of vascular bundles. Now, these are the main differences between the structural organization of root and stem, which you could summarize in a tabulated manner. Now, these are some questions where you can go through them and try to answer them. How many of the following features are exclusively seen in angiosperms with respect to vascular tissue system? Presence of xylem vessels, presence of sieve tube elements and companion cells, presence of albuminous cells and sieve cells, presence of tracheid and sieve cells. So angiosperms, they have xylem vessels, they have sieve tubes and companion cells. So the answer will be A and B only. Then the second one is this. Endarch and exarch are the terms often used for the describing the position of primary xylem in the plant body. And exarch condition is the most common feature in the root xylem. So you could try to answer it, whether it is endarch and exarch are used for the primary xylem or for secondary xylem. You could read and just compare and go for the answering nature. Then now this is the match the following, where you have bean-shaped guard cells, specialized epidermal cells around guard cells, dumbbell-shaped guard cells, and regulate opening and closing of stomata. You could match them with monocots, guard cells, dicots, and subsidiary cells, and you can actually get the correct answer. Trichomes are epidermal structures of root, appendages of pericycle of stem, epidermal outgrowths of dicot stem, hypodermal structures of dicot stem. So what are trichomes? You can read the 4 options and try to answer them. Then the next question is, root hairs are they uniseriate, multicellular, unicellular, or multiseriate? Which term is applicable to root hairs? Choose the appropriate one. Now the next one is the outermost region of the stellar ground tissue system and the innermost region of the cortical ground tissue system is respectively. So outermost region of the stellar ground tissue system you have to choose from the given options, and also the innermost region of the cortical ground tissue system. The options are, first one is hypodermis, second one is endodermis, third one is medulla, fourth one is pericycle. Now the next question is a statement type of question, where the statement A is, in dicot stem, pericycle is located above the phloem as a few layered semilunar patches made up of sclerenchyma. Whereas the statement B is, in dicot root, pericycle is next to endodermis, organized as few layers of thick-walled parenchymatous cells. So first statement is regarding the pericycle with regard to in dicot stem, which is made up of semilunar patches of sclerenchyma. Second is with regard to the pericycle in dicot root. You can read them and choose the correct option. Now this is another match the following question, where in column 1 you have the ground tissue of leaves containing chloroplasts, xylem and phloem together, vascular bundles with cambium, closed vascular bundles. In column 2, you have to match the column 1 with column 2 parts like mesophyll, vascular tissue, dicot stem, and monocots. Now this is the next. How many of the following features are relevant to monocot stem? So from A to D, you have 4 points. Just go through them and find out what are the relevant A to D points relevant to monocot stem and choose the appropriate one. Now radial vascular bundles are found in. Where are radial vascular bundles found? From the given options you can choose the appropriate one. Now the next question is, the non-stellar part of dicot stem that provides mechanical support to the plant axis is. So from the options you have to choose that layer which is not a part of the stele, because they have asked non-stellar part of the dicot stem which will provide mechanical support. One is epidermis, second one is hypodermis, third one is general cortex, and fourth one is endodermis. Then the next is medullary rays are present. Where are medullary rays present? So these are the 4 options where you can choose the appropriate one. Now the vascular bundles in dicot stem, what's the condition? Open collateral endarch, open collateral exarch, closed collateral endarch, closed collateral exarch. You could choose the appropriate one. And cortex is the region found between. So cortex is the region found between which regions? Isn't it epidermis and the pith, or what are the options that are given here. You can go through and find out the correct one. Now the next one is, in a dicot stem, there are few layers of radially placed parenchymatous cells between the vascular bundles responsible for radial conduction of water and minerals. Identify that tissue. So what is the tissue that helps in the radial conduction of water and minerals in dicot stem? So you have been given 4 options: pericycle, medullary race, pith and conjunctive tissue. The next question is, closed vascular bundles do not have. Closed vascular bundles do not have which part? Xylem, phloem, cambium or water cavities. So now, after going through this chapter, now try to answer these questions considering this diagram. During secondary growth, vascular cambium in dicot root develops from. So during the process of secondary growth, vascular cambium in dicot root develops from which parts? The answer for this is this. The vascular cambium during secondary growth in dicot root develops partly from cells of conjunctive tissue and partly from cells of pericycle by dedifferentiation. Now another one is, do observe the diagram and which type of vascular bundle is supposed to be shown in the diagram is not present in which transverse section. This type of vascular bundle is not present in which transverse section. So the type of vascular bundle shown in the diagram is not present in dicot stem, because dicot stem has open type of vascular bundle where there will be a cambium. That's the point. Hope this video is useful to you. Thank you.